In this talk, I'm gonna come up with a model to make predictions of the petrophysical properties of the matrix, because that's eventually where the long-term investment relies upon. And when you do direct experimental measurements of something as tight as shale, it takes a long time and it's very expensive. So having a reliable model is quite appealing to have. And we went on a quest in finding such a model. We looked at a boatload of SCM images of shale, and we basically realized three patterns. The first pattern, this could be represented by a single scale network model. So either you have an extrabasinal particulate sediment, or you have a large amount of organic matter pores that have organic matter material that have pores in them. In the second pattern, the smaller scale pores are in clusters, and they're in adjacent to larger scale pores, such as interparticle pores. And the cluster is acting in series with respect to the adjacent pores. In the third pattern, the smaller scale cluster is acting in parallel with respect to larger interparticle pores. So based on these three patterns that we looked at, we came up with three network model types. They're three-dimensional, they're unstructured. The first one is single scale. In the second one, the smaller scale cluster is acting, again, in series with respect to the adjacent larger scale pores, and the cluster is a network itself. It's a microcosm of a network itself. And this is the three-dimensional visualizations of the parallel type. Now, although our work is based on images, one needs to emphasize the limitations of SCM images. They have a very small field of view, especially for something as heterogeneous as shale. So that's one limitation of FIPSM images, and it will be very interesting to have some sort of representation of this pore topology in a more larger domain. And if you want to use something that has that capacity, such as X-ray microtomography, then obviously the resolution is not enough. So that's what I'm going to talk about, how we are going to address this issue with the limitation of the field of view of images. And inevitably, when you try to quantify the pore topology, you sometimes encounter funny images like this, FIPSM images like this. You have these sporadic pores that are isolated. They seem disconnected. And before you want to go and quantify this pore connectivity, you want to answer whether these pores are actually disconnected or not in situ conditions, whether they disappeared during image acquisition, or maybe simply the resolution of even this SCM image wasn't enough to capture them. So that's something that we're going to address as well in this talk. This is a little cartoon of our workflow. We have these network models that are multi-scale, multi-physics. They attempt to honor the pore structure and fluid physics of each respective scale. And after having done that, we're going to simulate the macroscopic phenomena we're interested in, either single-phase or two-phase. Now, we can also use some of this macroscopic phenomenon to tune in our network models, to inform our models. One would be sorption hysteresis, which is measured in exergy conditions that can increase the field of view, or the domain of the network that we are trying to create. And the other one is in situ conditions, and that would be the NMR response, specifically the restricted diffusion that is measured by the NMR response. So these are the macroscopic phenomena that will be the focus in this talk. If you look at natural sorption hysteresis in applied, in the literature, there are four main classifications. Applied chemists have been working on these since the 20s or 1910s, and you have four main classification of hysteresis. They're based on size and shape. So the number one and number two are unimodal pore size distributions. And in the first case, the pore size distribution is narrow. In the second case, the pore size distribution is wide. In the third and fourth type, hysteresis type, this sort of behavior is linked to the pore shape, specifically a slit-shaped pore. And now that people are trying to characterize shale via sorption hysteresis, whenever they use this sort of a classification to make inference of the pore structure. And we said, well, okay, we will use our network models and just simulate natural sorption hysteresis and see how they look like. So for single scale, looks like a type 1 and type 2 hysteresis. That's fine. The second network type, where the smaller scale cluster of pores is acting in series with respect to its adjacent larger scale pores, we see a type 3 hysteresis. And that's pretty cool, because the classical understanding is that if I have slit-shaped pores, then I will have a type 3 hysteresis. Here we just use spherical pores, and it's actually the topology that is mimicking this behavior. And in type 3 networks, we see a behavior that is not really in the categorization. And it would be very interesting to look at rocks that have that pore net topology, make sorption and measurements, and see if we can replicate this behavior. Another observation is that the natural sorption hysteresis is sensitive to the network topology, and something else we toyed with was permeability hysteresis that I'm not showing here, and that was not the case. The sensitivity of natural sorption hysteresis is actually promising. Here we matched the simulation to some experimental results that Max Yu gave us kindly, and we see that the model is flexible, and again, it's a type 3 network model that's been matching these experimental measurements. It's not necessarily slit shape that is producing this behavior. But also bear in mind that this whole natural sorption hysteresis story is only applicable to pores smaller than 100 nanometers, because beyond that, the capillary condensation is not as relevant anymore, and therefore it needs to be looked at as a supplemental method, along with images and other measurements such as capillary pressure to incorporate much larger scale pores. So we go to the second part of the talk about using NMR in calibrating the network models. This is a two-dimensional NMR map. The y-axis is the restricted diffusion. The x-axis is the reactivity time, which is related to pore size distribution. The y-axis, again, it's related to pore structure. And what we tried to do first, before we jumped into calibrating the pore topology, was let's go back to the first. Let's go a little bit. Let's back up a little bit and see if we can come up with pore-throat ratios to see if those pores that we saw apparently were isolated, whether they are actually connected or not in in situ conditions. Can we use NMR to tell whether those pores are connected or not in reservoir conditions? And we just use regular simple lattices, three-dimensional. We implemented a continuous time random walk algorithm. The networks are infinite acting. And what we observe is that up to pore-throat ratios of 10, the NMR response is significant enough to detect the existence of those throats. So if, say, the pore size is 50 nanometers, then I can tell the existence of a throat down to the size of 5 nanometers in in situ conditions. And that's pretty interesting. We did the same spiel with throat length ratios. If my pore size is 50 nanometers, then I can detect its existence up to the distance of 10. I can tell it's the distance of 10 nanometers, whereas if they're 250 nanometers apart, then the NMR response will not be significant enough. Now, we haven't run enough simulations to come up with a cutoff value. And I'm trying to make this point here that using this continuous time random walk algorithm that we coded it's very time consuming. And if we were going to use this in these multi-scale, multi-physics network models, they're going to be very impractical to code and very costly to run. So we thought, well, let's use something more statistical. Let's dial it down a little bit and use weighing factors, the probability of a particle to move from pore A to its adjacent pore B as a function of pore-throat ratio and throat length ratio. And you basically start with a swarm of particles on one of the pores and see at a particular time how much of them have gone to the adjacent pore, and that would be the probability of a particle to jump from pore A to pore B. We did that. We have this weighing factor as a function of pore-throat ratio, but when we compare it to the explicit method, we see that the statistical method is overestimating. And we don't know why. And although it's tempting to just fudge our way through it and just define some sort of weighting factor here by matching the explicit method, we are still trying to understand why this discrepancy exists. Okay, we couldn't resist in also analyzing the effect of pore shape on diffusion. The traditional analytical derivation says that the reciprocal diffusion is a function of surface to volume ratio. Okay, well, all these pores have same surface to volume ratio. It's a sphere, a cube, and square pyramid. But as we observe, as the angularity of the pores go up, the corner effects become more significant, and the reciprocal diffusion increases. So whether this is significant in longer times, we're still trying to address that. And here you have funky-looking pores in shale. You have crescent-shaped pores. You have angular interparticle pores. So it's prodding us to look at this problem in more detail. In conclusion, natural sorption hysteresis can capture the pore topology in shale rocks if it has enough sensitivity and it has enough flexibility. Pore shape is detectable with 2D NMR maps. This can be of really interesting result in trying to infer the pore shape and simulating, say, two-phase imbibition property. NMR can detect pore-throat ratios as high as 10. That was something that we found. Right now, we're trying to validate our models. We're going to calibrate these network models with sorption hysteresis measurements and compare the network topology quantitatively with FIPSM images. And with that, I would like to thank the sponsor of this work and our collaborators. And thank you very much for your attention. I'll be happy to take questions. Yes?
What is the scale of the FIPSM data that you're looking at?
You mean the resolution scale of the FIPSM?
What is, with most FIPSM sets of data are, 10 microns by 10 microns by 10 or something.
Yeah, that's, yeah.
Recently, we've tried to go 100 by 100 by 100.
Mosaic by doing.
We have some data that you might be interested in looking at that's on that scale. It shows more connectivity than we had seen at the smaller scale.
I'd be very thankful if you discuss this further. Because we're actually on the hunt for some data to do verification. Yes?
If you were to mention a single value of diffusivity or permeability that corresponds to your calculations, what would it be?
You mean like a number for permeability?
Physical units in Darcy.
Well, this is a hard question because it depends on the pressure, the permeability. It has Newton diffusion and slip flow in it, but if I have to
3,000 psi Darcy. Yeah.
Okay, 1,500 nanodarcy would be a value. For diffusion, 10 to the power minus 10 SI units maybe. Don't quote me on this.